So, what is microscope? Can anybody explain? Can anybody tell 2 or 3 words about what is microscope? Anybody? Anybody? Microscope. Come on, you people have seen it. You stand up and then say. Which things? Okay. So microscope is basically an instrument, or you can say device. So sit down. So basically we see the objects which are not visible through naked eyes. Okay. Now we are going to study about the compound microscope. Can anybody say why it's called compound? Why is it called compound? It uses? Okay. So, it is called compound because it's not composed of single lens. It's 2 or more than 2 lenses are involved in this microscope. So, our discussion will be based on the basic principles, how it acts, and then the key parts and the proper and the do's and don'ts with the microscope, or how we operate it. Okay. So the microscope basically consists of 2 lenses. One is the objective lens and the other one is the eyepiece. Okay. Through the eyepiece, which is basically 10x, it's a fixed value, it's basically 10x, and the objective lens, there are different variants of it. We basically start from 5x, here we have 10x, then 45x, and then 100x. According to our need, we increase the magnification. This is the principle of how the 2 lenses act. First, the objective lens, it actually creates a real image. It creates a magnified real image, and then the eyepiece, we get an even magnified virtual image. So basically whatever we see is the virtual image. Now you people also have to learn about the different parts of the microscope, right? So this is a monoocular microscope. Here we have the binocular microscope, is the eyepiece. Then we have the body tube, is the revolving nosepiece. The objective lenses are actually attached to it, and we can revolve and we adjust the magnifying power. Then there is a part which is the condenser. Then the mirror base, it also you can say is the illumination center of the microscope. Then the inclination joint and the fine adjustment knob and the coarse adjustment knob. So far everything clear till now, or anybody has any kind of question? So let's proceed. We'll start with the mechanical part. Mechanical part is basically the part which basically holds the microscope. Okay. We start with the base. Everything in the microscope is based on it, right? Then we have the arm. It's basically a C-shaped portion. Let me show you. It's the arm, C-shaped arm. You can also say it's a C-shaped arm. And then we have the stage. This one is the stage. We put the specimen in a slide and we place it here. And with the objective and the eyepiece, we see the magnified image of the particular required specimen. Then move to the next part. We have the coarse adjustment and the fine adjustment. Now there comes the tricky part, and you people will find it difficult or may find it difficult. So the coarse adjustment is basically we use it for fast focusing or rough focusing, right? And then for more clarity, we use the fine adjustment knob. Let me show you. Here is the coarse adjustment knob and the fine adjustment knob. So first, when we place the slide and we start focusing, we first use the coarse adjustment. By it, we call it a rough focusing. We roughly just bring little bit clearer image of the magnified thing we want to see. And then by the fine adjustment, we make it more clear. Then comes the optical part. Optical part consists of the eyepiece, which is 10x, and body tube, which holds the eyepiece and connected to the objective lens. Then the revolving nosepiece, which has all the required lenses, objective lenses you want. And then basically the objective lenses, which are of 4 kinds. Here we have 4 kinds, like 5x, 10x, 40x and 100x. So now comes the illumination part. The illumination or the illuminator is basically the light source. Actually, in our time we used to use through natural light, there used to be a mirror, we had to adjust it. But right now the microscope we have here basically have a bulb. So you can say this is the light source. Then comes the condenser. What is a condenser? A condenser is actually a lens which focuses the light in the specimen you want to view under the microscope. This is the condenser. And there is a diaphragm. Diaphragm is basically the amount of light you want to view a specimen under the microscope. It has to be adjusted with the adjustment or the objective lenses you are using. You have to adjust the diaphragm and the whole illumination part. Now here comes the adjustment. Now can anybody say here the objective lens is 5x but the total magnification is 50x. Can anybody explain why it is like this? Can anybody? Yes, exactly. The eyepiece is 10x. So we just straightforward, we just multiply it. So the total magnification with 5x we basically see as a 50 times magnified image. So when you are basically using the 5x objective lens, you have to keep the condenser position in the lowest position. There is also a knob through which we can actually adjust the illumination part, mainly the condenser. We can adjust it. You people will be demonstrated here. There you people will be described about everything. So the condenser position is basically you have to keep it in the lowest position, right? And then the iris diaphragm is basically like a camera. You have to open and close, adjust the shutter. It's basically like that. So a iris diaphragm should be almost completely closed. Though it's completely closed, there is little bit of like opening through which little bit of light comes. So at the lowest magnification, you will completely close the iris diaphragm. And coarse adjustment, mainly we at the lowest setting we use the coarse adjustment. As we magnify it, as we come towards 100x, we basically use the fine adjustment. There is only one thing, one purpose of coarse adjustment in 100x. It's breaking the slide. If you want to break the slide, you can use the coarse adjustment. So then comes the 10x. 10x is you have to keep the condenser little bit above 5x. Where you have placed in 5x, you have to keep the condenser little bit up, and you have to slightly open the iris diaphragm. In 45x, it should be midway between 10x and 100x. In 100x, you have to bring it completely up. So in mid position, or it will be according to your adjustment or how you are comfortable. And at the 100x, it should be completely up and the iris diaphragm should be completely open. So this is basically how we use the microscope. In 100x, we use oil immersion. Okay, we use the cedarwood oil in oil immersion. 100x is called oil immersion itself. because in 100x we used to decrease the refraction of the light as much as possible, because when the light basically comes from the condenser, it has to cross the lens and then is to come to air. So for that purpose we have to put oil so that it can be reduced to some extent. So can anybody name the oil? I have just mentioned it. Now comes the precaution, the don'ts about the microscope. So the microscope is basically, you can say it is a celebrity. It doesn't want to be touched by you. You have to always use a silk cloth to wipe it off and clean the microscope. Secondly, when we use the highly magnified image or oil immersion, we do not use the coarse adjustment, right? We do not use the coarse adjustment. And handling the microscope, it should be with both hands. One hand holding the C arm and one hand touching the base. This is like giving respect to the microscope, because you have to eventually break your head with it at the end of the year. So I hope you people have understood. This is all briefing about the microscope.